So it's, of course, very intimidating to give this named lecture. I think it means that I have now peaked, and so from now it's just downhill. So looking forward to that the next 20 years. So thank you very much for the very kind invitation and that very nice introduction. Before I talk about what I want to talk about, I would like to spend a few minutes telling you about my relationship with G.I. Taylor. G.I. Taylor, if you talk to someone from the community of fluid mechanics, which I encourage you to do, if you ask them to tell you who their hero is, not a politician, not a movie star, but their actual hero, scientist, chances are this is the person who will be number one on their list. He has inspired generations of people in my community, probably more than anybody else in the field. And in fact, this famous portrait, this painting, is in my building just a few meters from my office. So this afternoon I went and I took a picture of it. So this is at 4:01 p.m. today. This is convenient because this picture is just next to the coffee machine. So every day when you start your day, and you're waiting for a minute for your coffee to be finished, G.I. Taylor is looking at you. And, okay, so this is, you have to be, it's very intimidating. It's also exciting. So you have to measure up somehow to him. Now, if you're not from the community of fluid mechanics, I know there are people in the audience who have made a career working in flows and fluids. There are people in the audience probably who have never worked in fluid mechanics. It's very hard to convey how present he is when you, in our education, when you teach fluid mechanics to students. If you open a textbook in fluid mechanics, he's everywhere in the index. So we have Taylor vortices, Taylor columns, we have Taylor cells, we have Safman-Taylor instability, Rayleigh-Taylor instability. We have Taylor-Quette instability, Taylor-Quette cell, just many, many, many mentions of Taylors everywhere in the book. So he's really, really omnipresent. Now my relationship with him, when I realized who Taylor actually was, of course, you hear the word in classes, but you don't really, when you're young and you're being taught the field, you don't really link one Taylor from another Taylor. Now when I was a PhD student, I took a class, this is the only class I've ever taken, which was designed around one single human being. So this is a paper that came out before my PhD, Physics Today. It's actually free. You can download it online. And this was the basis of a course. And G.I. Taylor did so much in so many different fields, from fluid mechanics, solid mechanics. He did some thermodynamics, a lot of dimension analysis, and basically mixing all these things together, that there is enough in his own career for an entire syllabus in classical continuum physics. So I really encourage you to check this paper out. It's very inspiring. So the title of my talk today is Life in Moving Fluids. This is a title that I took from another place. This is the book. This is a book, another piece of scientific writing that I thought was very inspiring. I read this after the end of my PhD. This is a book by Stephen Vogel, who was one of the pioneering biomechanics professor. He was at Duke in the US. And he wrote this book, which is basically a kind of a journey through time scale and length scale within the biological world, telling you all the moments where fluid flows, and the emphasis is on moving fluids. Not just the fact that there is a fluid that doesn't move, but the fact that there is motion and there are forces associated with this motion, how that transport affects basically life, from the smaller length scale to the larger length scale. So this is what I like to talk about today. I won't talk too much about my work, just a few minutes here and there, but mostly I will be telling you, if you don't know about this field, I apologize for the people. There are at least two people in the audience who know everything about this field. I'm looking at you, Tim and Ray. I'm sorry, you already know everything about this. For everybody else, this will be a little bit of math, a little bit of physics, and a little bit of history, and I hope that at the end you will have enjoyed it. So, fluid flows in biology, they're relevant on basically all length scales, from cells and inside cells all the way to the largest organisms. And one of the things that make them interesting is that they involve interactions between, well, the fluid itself, but also active tissues, things that move from the biological world due to the source of energy. So there, if you look at a textbook, they'll tell you there are two types of fluid mechanics in biology. There are flows which are internal to organisms. Obviously, the famous one is the blood circulation. This is blood, so this is liquid. There's also an internal gas flow in your lungs, and that involves gas exchange between at the red blood cells level, then oxygen and CO2. And so this is another example of fluid mechanics problem inside an organism, except it's not a liquid in this case, it's gas. On smaller length scale, this is an example of an internal problem. This is something called cytoplasmic streaming, which Ray has done a lot of work on. These are motion that occur inside cells. So sometimes when you look at cells under the microscope, they look completely disorganized, random motion, and sometimes the motion look to be totally well organized, coherent. This is an example of such motion. This is a large plant cell where you have kind of coherent motion and transport of chloroplast inside the cell. Flows could also be outside things instead of being inside. So obviously flows around plants, flows around, this is a simulation in Germany around red blood cells and platelets. These are experiments and simulations of vesicles, single vesicles, lipid bilayers that are advected by flow and sheared and change their shape. And the quintessential external flow problem is the problem of locomotion, which I'm going to talk about today. And locomotion occurs from the very small length scale, microns, at the single cell level, all the way to, of course, things that move in a liquid, like a fish, also things that move in air, like a bird or an insect, and something in between. So this is, turns out, maybe it doesn't look like it from where you stand, this is a fluid mechanics problem, because there is a foot and there is mucus, and that's the mucus, fluid mechanics actually, and the rheology and the mechanical properties of the mucus that allow the snail to move. So today, I'm going to talk about some aspects of locomotion, and G.I. Taylor, by the way, is going to come in this talk 3 times for actual reasons. You will have to talk about his contributions. So this is a quick summary of the tree of life, origin of cellular life about 4 billion years. I should point out I am not a biologist, so all of these things I've learned from reading and talking to people. So if you ask me a biology question, we'll quickly reach the end of knowledge from as far as I'm concerned, but I can pretend I know some things. And today, the biological world is split between prokaryotic cells, like bacteria, and eukaryotes. So we are here, we're animals. And those numbers are the approximate number of different species between each of those categories. Microorganisms, very small things, actually contribute the bulk of the world biomass, more than 10 to the power 30 cells. Most of them are bacteria. And the motility, the ability to move around and as a result in a fluid to propel yourself, is shared across all of those. Single cells or unicellular organisms versus multicellular larger cells, prokaryotes on the left and eukaryotes on the right. And so even though there's a very large, very wide diversity within this biological classification, the physics, that's what I would like for you to get out of this. The physics is actually, a lot of things are in common. Even for things that have separated on the evolutionary tree almost 4 billion years ago. So this is something that's quite remarkable. So how do we think about things that move in fluid? Well, the first thing you do when you're a fluid mechanician and you're finished thinking about Taylor, the second thing you do is you think about Reynolds and Reynolds numbers. So the way to introduce the Reynolds number for locomotion is to imagine I have a body suspended in a fluid at a typical size L, and that moves around at a typical speed U. It's in a fluid, so the fluid has a density, there's mass per unit volume, and it has a viscosity, something about the friction of the fluid. For example, honey has very high viscosity and water has very low viscosity. So if you ask yourself, when I swim, what are the typical timescales that govern the dynamic of the surrounding fluid? Well, there are typically 2. One is an easy one and one is a slightly harder one. The easy one is the timescale to any perturbation. If I sit on the swimmer that moves, the timescale for any perturbation should just be advected downstream from where I stand. That's typically the size of the organism divided by the speed, so that's just an advective timescale. That's easy. The second one, which is a diffusion timescale, you have to know a little bit of fluid mechanics to know that if you perturb the fluid, a fluid that has viscosity, then that perturbation is going to be dissipated away, going away diffusively. And therefore, a diffusive timescale is a square of a length, the only length you have here, divided by a diffusion constant. And in this case, the diffusion constant is what we call the kinematic viscosity, which is mu divided by rho. So that is a timescale. That is a timescale for any perturbation to going away by due to the action of viscosity. Now, of course, if the thing is very viscous, the timescale is very short. So if you perturb honey and you start perturbing it, very quickly the motion stops, whereas if you perturb water, then it takes a while for the water to stop moving. So if I have 2 timescales and I divide them, then I get a number that has no dimensions, which is this one over that one, and that's the Reynolds number. Now, Reynolds is associated with another experiment. This is the picture from his paper. He was what is now the University of Manchester, created this very famous experiment, the transition from laminar flow, very nice, smooth, unidirectional flow, to a turbulent flow. These are his sketches from his paper. These are actually pictures from experiments that are reproducing this a little bit. There's a workshop right now at the Isaac Newton Institute on turbulence and wall-bounded turbulence. This is a kind of the classical hard problem in classical physics. And so this is the same Reynolds, and he proposed that there were a combination of all the parameters of the problem, only one that seemed to govern the transition from a laminar to turbulent, and it's the same thing, the one I'm talking about for locomotion. So if I go and actually look at the different organisms on length scale from a small bacterium, just a few microns, all the way to the largest thing I can find, a sperm whale, tens of meters to just a few microns, and I calculate the Reynolds number because, well, let's say they all move around in water, so I know the density, I know the viscosity, I know typical swimming speed, I know the typical size, so I can calculate the number. And it turns out there's a nice and clean divide between things that are very small Reynolds number and things that are very large Reynolds number. I'd like to point out how amazing it is. There's 11 orders of magnitude between this and that. There's a huge range. And the physics in this part of the flow is very different from the physics in that part of the flow. Okay? That's the divide between low and high Reynolds number. And I'd like to spend a few minutes to tell you what kind of give you intuition of what I mean by the physics is very different. So simplest way to have intuition about this is to think of a simple thought experiment. Imagine you were swimming, this is you, or a version of you. You're at the pool and you're swimming around, and then suddenly you're instantaneously tired and you stop swimming. So you know that if you go swimming and you do this, and eventually and you stop, you're not going to instantaneously stop. You're also not going to do 50 meters. That would be too easy. You're going to move by that much, okay, or this much, depending on how aerodynamic you are. Okay. So I'm going to ask that question. Can I estimate that length scale by which I move until I actually stop? So imagine this is a swimmer, typical size L. It used to swim around at speed u. It has a density, so it's mass per unit volume, which is rho s, s as in the swimmer. And I'm going to ask, how can I estimate the distance d that I will move around, that I will coast inertially before I stop? So that, of course, is set by Newton's law that says that I will decelerate because mass times deceleration is equal to the drag force from the fluid. The fluid is what's stopping me. If I'm in honey, of course, I'm going to stop very quickly compared to if I'm swimming in the water. Now, where the Reynolds number comes in is that this force, the deceleration from the fluid, the force, drag force from the fluid, critically depend on the value of the Reynolds number. So let's do this scaling. So I used to go at speed u, and I'm going to say that it will take me about the time tau to decelerate. So that means that I'm going to replace this dv dt, this deceleration, by the typical speed over the typical time that I will stop. So therefore, that relationship becomes an equation for tau. Tau is mass u over f. Now, if I know the time, and I used to go at speed u, then roughly speaking, that means that I will move an amount d, which is u times tau, roughly speaking. So therefore, my distance that I'm trying to estimate will be that thing times u. Therefore, it's m u squared over f. So far, I have not invented anything magical. Okay, I'm just following the logic. Now, the mass of something is the density times the volume of the thing, so the cube of the length. So I'm going to replace this, and I'm therefore going to get the final scaling, which is density, which I know what the density is. The length per cube, I can just measure. I know the speed, I can also measure in the experiment, and then I need to know f. If I know no f, then I'm done. Now, at a high versus low Reynolds number, f is different. So at high Reynolds number, the typical force, this is simplistic, overly simplistic, but let's say at the simplest way, I will say the force is the surface area times the typical pressure from the fluid. This is called a Bernoulli pressure. Of course, if the body is very streamlined, then the drag force is a little bit different, but let's imagine this is something that is totally not been optimized hydrodynamically, aka, say, me, when I go swimming. And that's going to be a good estimate for the typical order of magnitude of the force. Now, I know this formula. If I divide by that f, then I can calculate d. And it turns out that d over L, so that if I calculate d in units of the body, then d over L is simply given by the ratio of densities. So that's the estimate at high Reynolds number. So when we go to the pool, we are all, I hope, high Reynolds number swimmers. Otherwise, we're really bad swimmers. And our density is about the same as water, so this is 1. So that means that if I go to the pool and I swim and I stop, I'm going to move by something which is in the units of L. Say half me, or twice me if I'm very well optimized, but not 50 times me, or not, 0.001 me. Okay, that's the order of magnitude. By the way, if I'm not in a fluid, if I'm in air, then the density, say, of a bird relative to the density of air, that's going to be 3 orders of magnitude. And so that's why, if this is 1,000, the bird can flap wing a little bit and then continue for a very long time without having to do some more propelling, because it will take a long time for the air to stop the bird. Now, of course, the bird can suffer another consequence, which is to fall down due to gravity. So I'm not discussing lift and the issue of lift, I'm just discussing the issue of drag. Okay? If you go to the pool, you're not able to do this. Swim 2 strokes and then wait for the inertia to carry you all the way to the end of the pool, unfortunately. Now, at high Reynolds number, the scaling, at low Reynolds number, the scaling is different. The scaling is viscosity times speed times size now. To know this, you need to have taken a course in fluid dynamics, which I encourage all of you to do, and that's what you will get with the prefactors. Now, if you replace that formula F by this, then you'll get the scaling for the length scale. And now the length scale becomes the ratio times the Reynolds number. So what used to be 1 at a high Reynolds number now becomes Reynolds number. Now that number, I told you, is very, very small. This ratio, rho s over rho, is, say, 1. So this ratio of length scale is on the order of Reynolds number. If the Reynolds number is very, very small, that ratio is very, very small. That means that you cannot use inertia whatsoever. If I look at the bacterium, length scale is about a micron. The Reynolds number is about 10 to the minus 4. So that ratio, that means that D is about 1 angstrom, which means on a continuum point of view, it's 0. So the bacterium moves, and as soon as the bacterium says, that's it, I'm tired, stop moving whatsoever, instantaneously. So this is a world, it's not our world, this is a much smaller, much more viscous world, it's an instantaneous world. So here's an interesting consequence. This is a movie from G.I. Taylor. So G.I. Taylor was part of a series of movies done in the 60s, an initiative of MIT. He was responsible for the viscous flow, low Reynolds number flow. And he showed in this movie an interesting consequence of things that are very viscous. So being at very small length scale is the same thing as being very viscous. And he showed that if the flows are what he called kinematically reversible. So what he did is he had what is now known as a Taylor cell, basically a flow between 2 cylinders, and he put a little dye, injected a dye, and then he's going to rotate 1 cylinder with his finger. So look at where the fingers are, so that we're not cheating. So the fingers are pushing, and the little dye was dispersed, and now you can see it's being stretched. It's basically disappeared to never to be seen again. Okay, so the fingers are pushing. At some point, the finger is going to hop on the other side. Look at the finger. Okay, now it's on the other side, very important. We're not cheating. It's not the same movie played backwards. Now the finger is pushing the other way, and you should look at what happens here. So the piece of dye that was stretched, it basically disappeared, has a magical property, and the dye is coming back exactly where it started. Okay? Of course, there's a tiny bit of blurriness around it because it's diffusing very, very slowly, but because it's very viscous, it will take a long time for this to diffuse. So the flows, this is what it feels like to swim at low Reynolds number. Things you do will be cancelled out by the other half of what you do if it's reversed compared to the first one. So you have to just be a little bit careful. One consequence for locomotion is something that is now known as the Scallop Theorem. The Scallop Theorem says that if you're trying to swim, if you're a cell and you're trying to swim, but the motion that you do is time reversal symmetry, has time reversal symmetry. That is, if you were to film it with the camera, play the movie forward and play the movie backward, and those will be the same, then you cannot swim because the flows are reversible. Whatever proportion you're hoping to generate in one half will be exactly cancelled out in the other half. Okay? So those types of swimmers cannot swim. An example of swimmers that cannot swim are swimmers that have 1 degree of freedom. So for example, you know that if you've done any scuba diving, and if you've done this with your fins in the back, you know you're moving forward, right? But that would not work at low Reynolds number, because at low Reynolds number, every time you go up and then you go down, you're cancelling out exactly the amount of going up. So if you try to do scuba diving in honey, it is not going to work, okay? I do not recommend doing that. So this is an example of 1 degree of freedom, just a hinge, a single 1 degree of freedom hinge. You can have an example one, you can do mathematically very nicely, just 2 spheres. So you need more than 1 degree of freedom. So you can do this with 2. So there are very nice studies that I've done that it's shown that if you do it with 2, and if you actuate the 2 degrees of freedom very precisely in phase with one another, you can create motion, you can create forces. So that if you have 3 spheres, and if you separate those 3 spheres in such a way that is not time reversible, then you will get force. Purcell, from the Scallop Theorem, had an idea for a 2-hinge swimmer instead of a single hinge, and that also works. Now, the biological world does not have discrete degrees of freedom, it has continuous degrees of freedom, and the answer of biology to the problem of the Scallop Theorem is to create continuous deformation in the shape of waves. So on the left here, I'm going to tell you a little bit about how those waves are generated. You have bacteria, you have helical waves. Clearly, you can see in the movie time progressing from one direction to the next. Here, for eukaryotes, you have traveling waves, flexible waves. Clearly, also time. The wave is propagating in one direction. This breaks the symmetry. If you were to play those movies backwards, they will look like not the same waves. They will look at waves propagating the other way, and therefore, they are avoiding the constraints of the theorem. So there's a variety of swimming microorganisms, from prokaryotes to the left to eukaryotes. In eukaryotes, there are also a very large range in length scale, but what they have in common that is beautiful is that basically the physics of their motion is very different. Even though the mechanism that is responsible for the actuation, what is actually generating the force from a biological standpoint, is very different. So for bacteria, so I will take a minute to tell you about how exactly the geometry and the force is generated. For bacteria, prokaryotes, this is the analogous, this is a model organism, E. coli, and this is analogous to essentially kind of like a submarine. So if you have a submarine, you have something you want to transport, and you have a motor, and you have a propeller. The propeller are blades, and the motor rotates the blades, and that moves it forward. Here, the propeller is not a blade. A propeller is what we call a flagellar filament. It's the shape of a helix, but it's still a passive, kind of semi-rigid object that is rotated, just like your blades of your submarine. The motor here is something which is a rotary motor. It's basically the only wheel in biology. It's embedded in the body of the cell, and it's attached to the cell body, so the axis of the motor is fixed in the frame of the cell body. And because the flagellar filaments has to be able to move in all directions, you need to somehow transmit a fixed rotation in a particular axis to any other direction, and this is done by something called the hook. So there's a very floppy piece of polymer called the hook, and the hook transmits this. It acts as a joint, as a universal joint between the motor and the flagellar filament. So the motor rotates, that rotation rotates the hook, and the hook rotates the flagellar filament, and the rotation of the flagellar filament pushes the cell forward. Okay, that's a simple rotation. And this is purely passive. If you were to cut this in half, that second half would just be nothing. It would just not move anymore because it's no longer the actuation at the end. Eukaryotes are very different. They also use something called flagella, but the flagella are active filaments. And those active filaments have actuation which is distributed all around along the filament. So instead of having one point that is rotating, everything else being passive, the actuation is distributed everywhere, and it's very similar to a muscle. So a muscle have fibers which are sliding past one another to the action of molecular motors, and those motors are distributed along the fiber. Here is very much the same. Inside this structure here are molecular, long filaments called microtubule doublets, and those microtubule doublets all slide past one another, and if you add sliding with geometry constraint, then you'll get bending. So if I'm trying to slide this hand relative to this hand, then of course I can, but if I attach my hands to my shoulders, which I hope they are, then if I do sliding, then I'm going to get bending for free, because they cannot move relative to my shoulders. So if you combine sliding with geometry, you will get bending. And so this is exactly, this is exactly how this works. This is an active structure, distribution of basically active force all around the flagellum of the cell. And if this time you're able to cut this in half, as long as the two pieces of flagella still have access to nutrient, they will continue to beat on their own. So this is both active material. Right, so I would now like to convince you that even though they're very different and they've evolved from an evolutionary point of view on very different branches, the physics that they exploit is very similar. So I'd like to zoom in on both bacteria and eukaryotes, and then show you that they're basically the same physics. So if I zoom in on one of those flagellar filaments, this is a helix. This is a zoom of one of those helices, and that helix is rotating. So let's imagine that it rotates in this direction. So if I look from this side, it rotates in a counterclockwise, looking down at the structure from where I stand. If I look at locally a slope of the helix which is going up, this is a point in the foreground, that's the point that's close to you, then I can see that that point has a slope that is going up and is instantaneously moving down. Okay, because it's going down, it's going to disappear on the other side. Whereas this portion of the helix which is in the background has now a negative slope, but it's instantaneously going up. Okay, so let's remember this for a minute. Let's put this on the side. Let's look at what happens for eukaryotic cells. So if I look at a traveling wave, this is a traveling wave of my sperm cell. This is a sea urchin sperm. Obviously, any given material point on the wave, nothing is moving. It's just going up and down, but it's the distribution and the phase between different points that give rise to this apparent wave. So if I draw my wave, let's imagine I draw this wave at time t1 in the past, where those two slopes are highlighted, and then I draw the wave slightly later. The wave is moving to the right, so therefore my wave looks like this, the second one, at a later time. And my material points are only all just allowed to move up and down. So that means that the points on the slopes, on the slope which is going down, are actually moving up. You see there's been a displacement. This space means that there's been a displacement up, whereas those points which are on the positive slope are actually going down. So that means that in both cases, it's exactly the same geometry between motion the same relationship between motion and geometry. If I have a negative slope, I'm moving up. If I have a positive slope, I'm doing down. This is done differently. One is done in 3D through a rigid body that is rotating. One is done in 2D through something that is flexible and active, but it's the same relationship between geometry and dynamics. So why does that work? Well, it works for two reasons. One, at very low Reynolds number, the drag is linear with the velocity. So if I move in one direction and then I double my speed, then I will double my drag. That's true in any direction that I move. Okay, it's linear. The second reason why it works is because those filaments, the flagellar filaments for bacteria or flagella for eukaryotes, are slender shapes. They're not spheres. They're actually elongated structure. And for elongated structure, the drag is what we call anisotropic. It is still linear, but it doesn't have the same value if you have a shape. I don't have an elongated shape. Let's say this laser pointer. If I drop this laser pointer in an experiment and I measure the speed at low Reynolds number at which it moves down, it will go slower than if I drop it in this direction, because there's more drag per unit speed here than that. And that was also illustrated by G.I. Taylor. So G.I. Taylor, same movie, just 10 minutes later in the movie. By the way, that movie is on YouTube and also on the MIT website. You can, tonight, instead of Netflix, you can, G.I. Taylor. So this is an experiment where he drops a rod, a vertical rod and a horizontal rod. See, he drops the vertical rod at this point. It's behind, but because the drag is less on this one, it's the same weight, so they're subject to the same force. So therefore, the speed will be different. And you can see there's a race between the two, and the rod that is for which the motion is tangential, kind of aligned, parallel to the shape, is going faster. In fact, at that race, it's going to beat this one. And the ratio is about 2. There's approximately, well, exactly, but roughly speaking, twice as much drag perpendicular than parallel. Okay? It's a very simple experiment you can do at home. To work, it has to be very viscous fluid. So really, you need actually honey, or something like this, or oil. So the reason why this gives rise to propulsion is the following reason. Now imagine I take a piece of filament, and a piece of filament, it is a portion, a local slope of any of those biological shapes, is moving at some angle. In this case, let's say vertical. Well, what I'm going to do is first, everything is linear and instantaneous, so I'm going to project that speed with a component parallel to the filament and a component perpendicular, u perpendicular. Then I'm going to apply my linearity. So I know that I have a force, a drag force that resists u parallel in the opposite direction. I have a drag force which resists u perpendicular also in the opposite direction. But the coefficient between f perpendicular and u perpendicular is higher than the one between f parallel and u parallel, because this ratio of drag, the drag is not isotropic. So here, I'm going to say it's twice as much. So I'm going to make the arrow twice as big. And therefore, the total force, which is the sum of f parallel with f perpendicular, this is f, is not aligned with u. That's the key thing. So most of it is obviously in the minus u direction. So you move one direction, you get a drag, the component, most of it is put behind you, kind of resisting the motion, but you pick up a component perpendicular to you as well. So you move in one direction, and while you're moving in the one direction, you're feeling a force perpendicular. And that's the propulsion. That is the amount of force that you generate that actually is going to be useful for the locomotion. Okay? And all that I needed is to use linearity and use the fact that this is anisotropic. Okay? How anisotropic? Well, will just tell me how much bigger this arrow is compared to that arrow, and therefore, ultimately, how big green is. And by the way, if it was not anisotropic, if the drag was the same, then I will add up this blue arrow with just the first one, and it will be exactly in the minus u. It will be pure drag with no propulsion. Now, this is the calculation I just did. Of course, you can flip it. If you flip everything, if you flip the geometry, and if you flip the direction, then everything is flipped except the propulsion. The propulsion still goes to the left. So this is the key of why this works, is because if you are a helical flagellar filament and you rotate, at every single point on the filament, you generate propulsion through the rotation and the fact that you are a helix. You generate a propulsive force that is constant, that is always at every point directed in this direction. Okay, so if I'm here and I rotate a helix, that a helix is going to, like a corkscrew, is actually going to be attracted in some sense by the fluid. It's going to pull itself to the left. Now, eukaryotic cells are the same. The only difference is that for a helix, that propulsion arrow is constant magnitude all around, except here, obviously, because the amount of speed that I get depends on where I sit on my wave. The maximum propulsion is in the middle, and then at the very crest or bottom of my wave, I get zero propulsion, and then I do it again. So the propulsion is still always the same sign, but it actually is modulated in magnitude. Right, so I think I'm about halfway, which is good. So now what I like to do is this is the end of my general background introduction. If you have worked in this field, I apologize. You can now wake up. I'm now going to talk about more recent work and more recent research and kind of hopefully convince you that this is an interesting field, convince you that there has been a lot of interesting math and physics that has been done, and also convince you that you should really read things written by G.I. Taylor right now. So first, I will talk about, I will highlight two different types of organisms, as I've done so far. Spermatozoa and bacteria. I have done myself a lot of work on bacteria, so I'm going to keep this to the end. And I first will talk about spermatozoa. Now, in terms of relative length scale, this is probably more accurate. So there's a factor of 10, roughly speaking, in difference in length scale. They're both very viscous, low Reynolds number things that move in a fluid. But it's important to appreciate that the length scales are different, and in particular, issues of noise. There are some issues that affect bacteria, which larger spermatozoa will not be affected by. So this is my third mention of G.I. Taylor, last one. There's a very classical calculation that G.I. Taylor did. So G.I. Taylor was not afraid of the Reynolds number divide. He worked on probably orders and orders of magnitude of Reynolds number to the left or to the right. And he wrote what is considered to be the first quantitative mathematical biology paper, and it turns out that it's on this particular field. So he had conversations with a very famous zoologist. I believe this is the home of the Department of Zoology, right? Somebody knows? Yes, okay, good. So probably everybody who knows, they're aware of the work of Mr. James Gray, Sir James Gray. And in fact, if you read that paper that he wrote in 1951, there are bits of the paper that are very relating conversations that he had with Gray. And in fact, the fact that Taylor himself did not at first, at least this is what I understand from the paper, believe that you could swim with no inertia whatsoever. But if you actually take the equations of motion in a fluid and you say the inertia is zero, so the Reynolds number is basically zero, exactly zero, then would the locomotion be impossible? Would you need a little bit of inertia to give you a little bit of locomotion? So he was not convinced, and so he had to do a calculation. That's in the G.I. Taylor, this is the G.I. Taylor motto: if you don't believe you're something, then you're just going to solve it. Okay? And he did this for 20 fields. So he said, okay, well, sperm cells, so James Gray worked a lot on cilia. They also talked about spermatozoa, and he said, okay, sperm cells have waves, so I'm going to make a calculation for a wave, but a real wave, for a real flagellum, is a three-dimensional slender object. That's very difficult. So it took him one more year to do the slender object. There's a 1952 paper that actually solved the real problem. In 51, he said, let me start with a simple problem. I'm going to do it in 2D. So I'm going to do a two-dimensional spermatozoon. Basically, it's a magic carpet calculation, except there's no inertia. It's a zero Reynolds number magic carpet calculation. So you have a sinusoidal wave. It's 2D. It has a wavelength. It has an amplitude. It has a wave speed. And you can actually do this calculation. You cannot solve this problem by hand in general. You have to use numerically. You can do it by hand, what we call asymptotically, provided the wavelength is large compared to the wave amplitude. Okay, so you do it in the long wavelength limit, and that is something we teach here in Cambridge. You can do this calculation. It takes about 20 minutes to do it on the board, and this is what he got. So this is what he got in his paper, and he actually carried out the expansion in higher powers of AK as well. You got the next, I believe, the next two terms, at least the next term. So the speed divided by the wave speed, which is the other relevant speed in this problem, is minus a half of the square of the dimensionless amplitude. So the actual amplitude times the wave number. Now, this formula is one of the biggest formula in this field, 1951, so this is quite a while back. It has many different things that are important. One, it's non-zero. So there is a non-zero speed, you can actually swim. Number two, it's minus. So if you send a wave in this direction, you're going to swim in the other direction. Okay, this is exactly what you see for those cells. And then three is quadratic in the amplitude. I mean, as it should, because by symmetry, you can't really have anything that would be linear in the amplitude, because the amplitude is going up and down, so that wouldn't make sense. But at least you get it that it's quadratic. It could be power 4, it could be power 6. It's actually the first kind of even power is where you get the motion. So that formula, versions of this formula, tens of different versions of that formula have been derived by many groups in many different topics for many different fields, including research in my group. But that's the original formula. And there's more in this paper. He does many, many more things. What would be now someone's thesis with 4 different papers or 4 qualitatively different things, for him was probably a Sunday morning, and that was this paper. Okay? I really encourage you to look at that wonderful paper. So now, later, so 1951, we're now in the 2000s, we can do much more. This is an example of an experimental group, joint experiment in theory, the group of Frank Ulicker in Germany, where they've done tracking of both sperm cells. Now, if you do this, those cells have planar waves, so this basically planar tracking. You have to track 2 degrees of freedom in translation in the plane, and then 1 degree of freedom of rotation of the cell if you just track the cell body. Cell body, of course, could translate in 2D and also rotate. And so this is measured using the 2 components of instantaneous velocity, the velocity parallel to the body and the one perpendicular to the body at each instant as a function of time, so of course it's periodic. And then 1 degree of freedom in rotation, so this is measured with the angular velocity of the cell. And they have a model, so the experiments are in black and the theory is in red, a very nice agreement, and the theory needs to have constraints to find those 3 degrees of freedom. You need 3 other constraints, and the 3 constraints are in the plane, I have no force in either direction. The organism is free from a force point of view to move, and there's also no torque or no moment. It's also free to rotate. So it's a free from a mathematical point of view, from a dynamic point of view, it's free organism. So there are 3 constraints for 3 degrees of freedom, and you get this beautiful predictive power of the theory. So now we can actually do more than the swimming sheet. I mean, it took 60 years, I guess, but now on your laptop, you can run simulations, and you'll be able to predict exactly what a cell should do. And in fact, we now understand how the cell moves, because you can see we can predict everything about this motion. We can compute flows. This is from a beautiful paper in the Annual Review of Fluid Mechanics. This Annual Review of Fluid Mechanics is a journal that comes once a year. It's basically a big review paper or state-of-the-art in a particular field. And 11 years ago, there was a paper on the hydrodynamics of sperm cells. So I highly recommend you look at it. On the top, there are experiments on, these are human sperm cells, and simulations, numerical simulations in a low viscosity fluid where you get very nice high amplitude waves that are generated all around the flagellum. And then on the bottom, these are in a very high viscosity fluid, and if you do this, then of course the forces in the fluid change, and that affects how the cell behaves, and so the waveform that is generated by the cell is very different. In that case, the wiggling motion of the flagellum is only limited at the end of it, the smaller amplitude, and in fact there is no motion here. And we now can also on the laptop solve for the flow. If you give me the kinematic of the cell, I can solve for the flow around the cell, and I can tell you, for example, what the velocity field as an actual field, time varying in space varying field, and the pressure and the forces, etc., etc. So we now know how to do this very nicely. Now you can use this to learn something about biology. So let me show you one particular case where you can do this. You can ask yourself, okay, this organism, this is from the same paper, this organism is moving around and is waving its flagellum because something biologically is creating a force. After all, we're not doing anything, we're just looking at it under a microscope, and yet it's moving. So I would like to know, what are the forces? Of course, those forces are periodic, because it's to the right, and then to the left, and then to the right, and to the left. But I like to know the distribution in space of those forces, and I like to know their magnitude, etc., etc. So how can I do this? Well, thanks to fluid mechanics and basically mechanics or continuum mechanics, we can do this. So here's an example of how you can do this. This has now been done by many, many groups. I'm just showing you pictures from this particular study. So these are human sperm cells. You can track the cell, track the flagellum, so use image analysis to actually figure out where the flagellum is at every instant of time over many, many, many times. And so you can reconstruct using image analysis the time series of the shape of the flagellum. Now, if you have the time series of the shape of the flagellum, you can then use a mathematical model, which I will explain. It's not very difficult, at least the concept of it, to estimate what the forces are. So purely based on observations, so literally taking movies, plus image analysis, plus doing some thinking about the mathematics of what actually is going on, you can actually know something about biology. You haven't really measured those forces. You measured something else, but because you know the principle of physics that the cell has to obey, you can get those forces. So the model that allows you to go from the image analysis to the time varying is this. So I'm not going to, of course, go through solving anything. I'm just going to highlight to you how this works. So I'm imaging the cell, and I'd like to figure out what is, at every instant, the force F, density. This is a force per unit length. So at every point on the cell, there is a force that is due to the fluid. So it's a drag force. Okay, so this is F, it's a vector, because at every point, you could point in three directions, and it depends on S, the arc length along the flagellum, and then, of course, I can do this at every instant of time. Now, we, the community, this is a classical result from 1980, we know the relationship between the speed at some point on the flagellum, let's call it U, and the distribution of force everywhere else on the flagellum. So it's an integral problem. If you know the F everywhere, then you know U. And the opposite is a bit tricky to do, but you can do it, is to invert this, is to say, if I take movies, and therefore I know U everywhere, I can numerically invert this integral relationship and get F. So the hydrodynamic force, the force due to the fluid motion on the flagellum, is something we can, with a computer, invert. So if you observe U as a function of space and time everywhere in the flagellum, you can get F. So you have to believe me. If you don't believe me, then that's it, we're done. But if you have to believe me to continue to the next step, it's okay? I hope you believe me. So we now have F. Now the flagellum is subject to mechanical equilibrium. There's no inertia, so at every instant of time, the forces on the flagellum have to balance. So I have the force from the fluid, which I told you I'm calling F, and that's, I told you how to do it. I have the forces from the fact that the flagellum is a piece of elastic material. If I bend it, and then I let go, it will come back. So there are elastic forces, but those elastic forces only depend on the shape. How much is it bending and how much is it twisting? So if I have a perfect image analysis, I can also estimate what the elastic forces are. And the third type of force are those biological forces, the fuel, the thing that actually creates the motion. That's the stuff I want to estimate. So therefore, if I have the movies and the image analysis, I get the U. If I get the U, I get the F. And if I get the shape, I also get the F elastic, which means I have this at every instant, those two terms, which means that at every instant I can estimate this. Because it has to, whatever it is, it better balance the other two. And I can do this at every time, and then I can reconstruct this. So these are examples of time-varying forces that are generated inside the cell. Not that we measure them, but because we measure something else, and we know that because of physics that has to be. That's the only way that it can be, that the cell is at equilibrium. This is an example where you use fluid mechanics and basically continuum mechanics in the spirit of Taylor, and you get access to biological things. Okay? The last thing I want to say about eukaryotes and sperm is an issue of optimization. So suppose I give you a microorganism that uses a flagellum, like say a sperm cell, and I want to think of I want to think of this as a robotics type problem, okay, an engineering problem. How should I deform my flagellum? What's the best way to do this? Okay, if you go to the pool, you know that there's a bad way to swim and there's a good way to swim. Okay, so that's the same here. What's the bad way for the cell to swim in the good way? Now there's a classical paper from Pierno and Katz in 1974 that said, well, if you give me a shape, instantaneous shape, take a picture, just a picture, not a movie, then the best way for the increment of that shape to move is through a traveling wave. This is a beautiful calculus of variations calculation. So it has to be a wave. For whatever shape you give me, it has to be a wave. That's the best way to move one small increment in time. Best in an energetic sense. The motion, you will get the most swimming basically for a given amount of power. So it's a traveling wave, so you can ask yourself, well, I know how to characterize a wave. I know how to parameterize waves. Of all the waves in the world, what's the best wave? So that's a calculation that Lighthill did on the pantheon of fluid mechanics heroes. Is number 2, just behind Taylor. He's also a Cambridge person. And he's shown that the optimal traveling wave for a sperm had a constant angle, about 40 degrees, between the local slope of the flagellum and the direction of motion of the wave. Okay? So you can do this calculation. We also teach this in part 3. Now, what's a shape that does this? Well, in 3D, it's very easy to make a shape that has a constant angle between the slope and the direction. That's called a helix. So a helix is optimal because it has a constant shape everywhere, constant angle between local slope and the axis of the helix. Okay, so that's the beauty of helices. In 2D, you cannot do this because in 2D, that means you will get kinks. And this is a problem you can regularize. Clearly, there's a problem with the kink. There's infinite elastic energy, but there will also be, in fact, infinite, at least locally infinite dissipation. So this is a problem you can rigorize. This is something that I've worked on. I think this is the first time I'm showing you stuff that I have done. This is regularizing this by taking into account the flexibility of the organism. So there's a new dimensionless number called the sperm number, which is basically the ratio between the hydrodynamics forces and the forces due to elasticity and flexibility. Now, if the sperm number is infinite, then you don't care at all about the elasticity. Then you get the optimal shape of Lighthill, which is this black curve. But then if the elasticity becomes more and more relatively more and more important, you get those shapes. And you get those shapes when sperm number is on the order of 1, then you get those shapes which are straight lines followed by circular arcs. If you plot the curvature is going to be constant for a very long period of time and then a big jump. So it's straight, and then it's circular arc, and then straight, circular arc. So that's kind of the optimal, from a mathematical point of view, the optimal shape for a sperm cell. And in many experiments, this is what they see. So these are classical papers from Charles Brocra, who was a pioneer working on invertebrates. And these are experiments on dinoflagellates, where he measured, this was in the 60s, all those studies, and he measured and cataloged a lot of different species. And he's observed that, in fact, many of the shapes that he sees are circular arcs. So he actually used image analysis. In the 60s, you have big tables cataloging curvature as a function of arc length, where a constant curvature and then there's a straight bit, and then a constant curvature and a straight bit. Okay, so it's kind of interesting that the mathematical problem, purely based on continuum mechanics and energy, is able to reproduce some things that you see in the biological world. So that's the end of my spermatozoa. I have 10 more minutes, so I will tell you now a little bit about bacteria, which is something that I've been working on a lot with my group over the last few years. I've already show you different pictures of swimming bacteria. There's a wide variety of ways to distribute the flagellar filaments, whether they're all over the cell body, whether they're all coming from the same point, but they all, as I said, try to emphasize earlier, using the same physics. Now, to think mathematically how bacterium swims, you need to solve, this is a simplified version of it. Imagine that I have a bacterium that only has one flagellar filament and that just swims forward in 1D. Okay, for simplicity. So it has a flagellar filament, it has a cell body. The cell body is going to rotate in one direction. Of course, because the motor rotates, we have to allow rotation of both the cell body and the flagellum, and the flagellar filament. So let's say it moves with speed u in 1D, one direction, rotates with angular velocity omega, and then because there's relative rotation due to the motor, the flagellar filament will rotate, but it will rotate at a different speed because there is a motor, and the motor prescribes a relative motion. So if this one minus this one is little omega, which is the motor rotation that is prescribed. So let's imagine that it's given. The motor is telling us how fast it rotates. Now I need to find two things. I need to find u, and I need to find big omega, and if I have, then I'm done. So therefore, I need two equations. I need to solve, of course, for the fluid mechanics problem, subject to two constraints. One constraint is that the forces have to balance. That cell cannot be subject to any net force, and the torques also have to balance. Now, to do this, I need to characterize the hydrodynamics of a helix. Now, this is something we do in part 3. I don't expect to derive any of this, but basically what you have is for a helix, if a helix translates and rotates, then it will be subject to a hydrodynamic force and a hydrodynamic torque, which is linearly related to u and omega with this big matrix. Okay, remember that the drag is linear, so therefore it's an instantaneous relationship between forces on one side and kinematics on the other. And if you solve fluid mechanics problems, well, you can solve for the value of the coefficient. That matrix has to be symmetric, so the B is the same off diagonal. And then you can calculate ABD. The C perpendicular and C parallel are the drag force per unit speed on the filament, perpendicular and parallel. And we can do this. It's kind of straightforward to do this for a helix. The important stuff is B, because the off-diagonal term says that this is links omega to F. This is the one term that says if I rotate my helix, it is subject to a force. This is the proportion that I told you earlier. So that B term is this one. It is proportional to the difference between the two drag coefficients. So if I have isotropic drag and those two drag coefficients are identical, boom, no B, no propulsion. And B is positive for a left-handed helix, which means that if I rotate omega in the negative sense, so viewed from the back, on this side, in the counterclockwise sense, it generates a propulsion to the right, which is exactly what you see in real life. Now, if I put this now on a cell, well, the relationship for force and torque on the helix I wrote over here, the helix, remind you, also translates with the same speed because it's attached to the cell and rotates with this angular velocity. And then the body only has a diagonal component because the body, let's say, is very symmetric. There's no coupling. It's just, basically a sphere or so the rugby balloon, whatever shape that is very simple and symmetric. And because the cell is force-free overall and torque-free overall, I better have that the force from the flagellar filament plus the force from the body have to sum up to 0, and the torques also have to sum up to 0, so I have a big linear system to solve. It's a 2x2 system. It's very easy to solve, and you can do it. So this is the solution to the swimming problem. It's not very difficult. All you have to do is you have to know how to calculate those coefficients of the matrix. So you know the swimming speed, U, proportional to the motor speed, the head rotation rate also, and the flagellum rotation rate also. And in real life, what do we observe? Well, in real life, say for E. coli, this is a left-handed helix, and omega is negative. The rotation is counterclockwise. If I go on this side, and if I look from the cell from this side, I see, looking down on the cell, I see rotation in the anti-clockwise sense. Then it predicts that U goes this way. So it swims pushed by the flagellum, cell body first. And the flagellum and the cell body always counter-rotate. So in the frame of the lab, if you were to be able to measure rotation, you would observe cell body going in one direction and the flagellum going in the other direction due to this balance of moment. And so therefore, this is the picture of what the cell does. The cell has a flagellum which rotates counterclockwise from behind. The cell body goes the other way, and then together, as they're doing this both rotation equal and opposite in some torque sense, then the cell moves forward. But this was for 1D, and in 3D, life is more complicated because now U and omega is now each vectors with 3 components, and because when you solve the force and torque balance problem, this is the body matrix, this is the helix matrix. In general, the instantaneous value of the speed as a vector of the bacterium will not be aligned with the instantaneous vector of rotation. They will be misaligned, and that means in general you get a helical trajectory. So the shape is helical, and the trajectory also is helical. And depending on what you have in your population, you can have a wide range of helical. So this is distribution for Bacillus subtilis of the pitch of the helix and the radius of the helix, of the helical motion itself. Now we can now calculate flows. This is an example of a numerical simulation done by a former postdoc in the group, Debashis Das, who's now in Strathclyde, is a lecturer in applied math. This is the flow that is instantaneously generated by a bacterium that has all the relative shapes and sizes for E. coli and has one flagellar filament. And obviously associated with this flow, clearly you can see rotation over here in one direction. You can see rotation. Well, of course, there are no arrows, so it's clear the direction, but just believe me that it's this way, in the other direction. And associated with this are also flows from the front and on the back. Why? Well, because as the cell is rotating its flagellar filament, it's actually pumping fluid backwards, so it's pushing fluid backwards. The cell is swimming, and so because it's swimming, it's also pushing fluids from the front. So as it's swimming, it's pushing fluids from the front and the back, and therefore, by conservation of mass, it has to attract fluid from the side. Now, this has been measured. This is a heroic experiment from Ray, who's sitting right here, more than 10 years ago now, where they've patiently waited, my understanding of the experiment, they very patiently waited for over long periods of time for bacteria to swim in the field of view of a microscope that was seeded with small particles so that they could measure the flow field right at the moment where the cell swims through, and then do it again and again and again so that they get beautiful statistics. And the result is remarkable. So these are the experiments for the flow. These are length scale much larger than the cell. See, the cell is not even 10 microns, and so this is the bar. This is the scale of the experiment. Right here it becomes very noisy relative to the actual flow that is created by the cell. But this is the measurement. This is the theoretical prediction that I just highlighted to you a minute ago. You can do this calculation. We teach this here in part 3. And it predicts that there's flows being created going away from the cell, front and back, and being contracted on the side, which is exactly what you see. And even more remarkable, it predicts that there are regions where there's no flow. There are actually critical angles where you can find cosine of an angle being exactly plus or minus a value, where it predicts there is zero flow, and those 4 regions are actually exactly seen in the experiment. So it's very remarkable that you get all those fine features of the flow in that very difficult experiment. And I emphasize these are just a few microns in length scale. The last aspect, and then I'll stop, that I want to highlight is the issue of polymorphic shapes. I told you that the flagellar filament was a rigid shape. It is rigid, but it has different conformation. It's a polymer, and the monomer called flagellin can actually exist in 2 different conformations, and that means that there are stable conformations of the polymer itself. There are multiple versions, and all of those, depending on which of the monomers and their alignment along protofilaments are in one state or another, there are 12 shapes that exist that are stable. And I believe out of those 12, 9 have been seen experimentally. And they've been numbered. Number 0 and number 11 are just straight, so they're not particularly useful for locomotion, because if you rotate something straight, nothing. And so there are 10 of them that are helical, and they all have different names. 3 of them are left-handed helix, so they rotate in one direction. The other ones are right-handed, so the chirality is reversed. And 2 are important. One is called the normal shape, because that's the one that's normal. It's used for actual swimming for wild-type cells. And semicoid is used for something I'm going to highlight in a second. And the other ones have also, some of them have also been given names. Now, why this is important? Because they're used for cells to change direction. So if you're in a cell, if you're swimming forward, there's something called, for many cells, called run and tumble, where a cell used to swim in one direction, and now it's at the end of the tumble, it will swim in another direction. But there is a mechanism by which it changes direction. So instead of steering the wheel, what it does, it rotates some of those motors, it flips the direction of rotation. That changes some of the flagellar filaments from typically normal to semicoiled, which then will basically give a kick to the cell, a random kick to the cell in a different direction. And at the end of the process, so this is called unbundling, when the bundle of flagellar filaments come apart. This is called bundling, when they're coming back together. And at the end, the net effect is the cell is the same 1 and 8, it's just been changed direction. So it's being kicked this to the left or kicked to the right. So this is the running motion in blue, and the tumble is this very quick 1/10 of a second reorientation of the cell. Now it turns out that process, so Masha, who's sitting right here, has done a study that has shown that process turns out is all governed all by physical mechanics. So if you do carefully the solid mechanics, which other people have done with the hydrodynamics and the hydrodynamic interactions about the cell, the unbundling stage where the flagellar filament come apart, the moment of the propulsion where you have combination of semicoiled filament pushing on the cell and normal flagella pushing on the cell, and then the bundling where it's coming back together. And if you put it together, you get the prediction. This is a totally deterministic calculation of the probability distribution function of the angle of orientation during a tumble. So the goal of a tumble is to swim one direction, and then at the end of a tumble to be in another direction. So these are the experimental, this is the classical Berg and Brown paper from 1972 in purple. Very recent paper in 2015 where we've redone that experiment. They measured long times and the population of E. coli, and the prediction with no fitting parameters is this blue curve, purely based on the consequence of physics and mechanics and their interaction of the flagellar filaments. Okay, so this is this paper by Masha last year. The last aspect I want to, and then I will be done, I promise, is the issue. Remember I told you about optimization of eukaryotic cells, traveling waves, and what's the shape of the wave. Well, you can ask a similar question about optimization, but about now shapes. Those shapes are, this is a much easier problem. There's no deformation, there's no wave. This is literally just building a slender shape, which is a helix. So you can ask yourself, of all the different helices that you could build, which one are the best from a hydrodynamic standpoint? Which one, per amount of dissipation in the fluid, or work done against the fluid, is going to propel you the most? And we can catalog all of them. We went to the literature, and we looked at all of them. There were many measurements, and we actually measured, there's a way you can measure the intrinsic efficiency of this as a propeller, as a function of the angle of the helix. So remember that angle is the angle between the local tangent and the axis of the helix. Of course, if it's 0, it doesn't swim. If it's 90 degrees, it's a circle. It also doesn't swim, so it has to pick somewhere in the middle. And the amazing result is that, so black are us going to the literature and looking at all the experimental evidence of measurements of shapes. Red is the theory of a theory that was predicted in the 70s by someone here in Cambridge, Caladine, who predicted what the shapes would be purely based on biochemical equilibrium. And the normal shape, this normal flagellar waveform, is the most efficient of all of them. So if you have to pick one for swimming, of all the ones that are stable, that's the one you should be picking. That's the one that are used. It's about 25% more efficient than the other ones. And the other thing that's interesting, I'm not going to spend a lot of time, but you could ask yourself, well, I don't want to get the best within the shapes that are already stable. Can I make an even better shape than the one that are stable? Well, it turns out you can, but you not gain too much. So here, this color map are other contours of this efficiency as a function of two parameters, the circumference of the helix, so the circumference of the tube on which the helix is coiled, and the wavelength of the helix. It turns out you have to lie on the circle purely by conservation of the amount of material. You have to lie on the circle, so the best ever shape you could build would be the intersection between the circle and the warmest region on this graph, which will be here. Okay, and real life is here. Okay, and so it's basically just a few % of the optimal that you could design in a computer. So it's remarkable from a purely, again, this is purely fluid dynamics, purely continuum mechanics point of view, that you get so close to the actual real-life optimal. So a lot of the things that I talked to you today about are actually, they're in this book. This has been many, many years of work. I don't know if many of you have written books, but this has been quite an intense experience every morning for basically 4.5 years. And the art, I'm very happy because the art in the book was done by my dad. So my dad was a biologist who worked on insect and then later on communications between animals. But he's also an artist, and he knows how to draw a lot of things. And all the book, the art was done by him. And so I encourage you to, not even have to buy it. You can go to the Cambridge Library, get a copy, and at least look at the art. And there'll be a little bit of math also on the side. Cool. And that's it. I would like to thank you all for coming, and I'll be happy to answer any questions.